flowers. It is the same species, so it shows variation. It either has white flowers or blue flowers. But when we have a crossbreeding between the white flower and the blue flower plants, all the offspring was blue. That helps us to see that this flower color is controlled by the dominant allele blue. My explanation is the F1 generation all shows only the dominant allele, which is blue. Use the symbols capital B, small b to represent the two alleles. So each parent was capital B, or small b, small b. And the genotype of the offspring will be one from there, one from there. Because during meiosis, gametes form and there's only one allele per gamete. And there we have the offspring. When we make a genetic diagram, when we cross one of the blue flowers with a white flower, the blue flowered offspring. So we use the heterozygous blue with the white. That is the phenotype. The genotype is capital B, small b, cross with small b, small b. The gametes will form during meiosis, and my gametes are in circles. Fertilization shows capital B, small b, small b, small b. The phenotype of the offspring is blue, blue, or white, white. So I can either have a heterozygous blue offspring or a white offspring. If they ask the ratio, this is the ratio. If they ask percentage, 1 out of 2 is 50%. Then we have the cop of a maize plant. And remember, when we draw a graph, this is X, this is Y. This is independent. We had a 7, a 8, a 9 cop. Then we count how many was there. That was 7 centimeter. 3 of the cops was 7 centimeter. 10 was 8 centimeter, right? And we draw a histogram. You will get a mark for having length of cop here and number of cop there. We did not sort this into ranges because it was a small amount that we were working with. We did not start at 1 centimeter and 100 centimeter. No, we had only 8 different. Shortest extreme was 7 and the largest was 19. Therefore, we could have written it like this. So you get for axis the name with its unit and the name with its unit on X, on Y. That is your axis mark. Then scale so that that distance is the same. And look at the 7, it's in the middle. Now, we write it in the middle now for the name of the column. Your column, you get your marks when they are attached, same width each column, and draw with a ruler. Attach because it's a histogram, no spaces. Draw with a ruler. That will give you your mark for columns and the same width. And then for your plotting, that was accurate. That is where you get your marks from there. We say it is a continuous variation because there's a smooth gradation, no distinct categories. Or we can say there are extremes with intermediates. From 7 to 19, there are all the different lengths of cop sizes. The environment can play a role. the amount of light, minerals, water, temperature will or can influence the size of the cobs. Explain how the type of variation shown by the maize cobs differ from that shown by the blue and the white flowers. They were discontinuous variation. You have to mention the name, describe it, or use the name to refer to it. And then we describe what do we mean with discontinuous. There's only blue or only white. No in-betweens, no intermediates. There's distinct categories. So I hope you got that answer. Grade 12. Now we can continue with our mutation lesson. Now that we have covered, there are variations. Variations is caused by the environment or by the genes. We have two categories, continuous. We start with one extreme, smooth gradation to the other extreme and all the in-betweens. Continuous, draw a histogram. Then we have discontinuous, distinct variation. Now, but in this variation, there can also form mutations. What is a mutation? A mutation is a sudden and unpredictable change in the genetic material of an organism. Now you remember we had three ways how a gene can change. A gene can change by if one of the bases is out, is deleted, or one can just be changed. It was a guanine, now it's a cytosine. So changed or deleted, altered or changed or deleted, or if a sudden an extra one appear. That is when the gene change. Now, and when the gene change, the order of the bases change in a gene, the recipe outcome of that protein will be wrong or different. The one mutation that we talked about was cystic fibrosis. You remember that gene that makes the mucus watery and not too sticky. If it's a faulty gene, it's a sticky mucus and prevent the uptake of oxygen. You got that one? Okay, that is gene mutations. And then we also have chromosome mutations, where there is something wrong with the chromosome in its completeness. So gene mutation, this sequence of the bases not copied precisely. And that changed the formation of the protein. Hemophilia, prevent blood clotting, that protein is wrong. Or albinism, where melanin, the part in the skin that causes the color of the skin, that brings color to the skin, but also protects the skin against the UV light, is not present. And then we have albinism, like we can see in this line. That is a gene mutation, which means either the gene is altered by having one or more of the gene bases changed, one is removed, or an extra base, nitrogenous base, is added. That's a gene mutation. We also add here cystic fibrosis. Now, is something that we can already see as mutations. We did that during our genetic problems. What about color blindness? It's also a defective gene on the autosome. The sex chromosome is also called the autosome. Now, that is also gene mutations. There we see a gene mutation where there's an overproduction of melanin. And here, very interesting, the two variants, overproduction of melanin and an underproduction of melanin. And it is not a new species, it is just a variation in the color. This is a lion, an African lion, but there's a variation in color, overproduction. And you know what happens if there is a lion like that? They all want to buy it because it's a very unique color variation. And if they start to breed with this male, there will be offspring that also look like this lion, like this male. So when we look at a chromosome mutation, it means there's a change in the structure of the chromosome or the number of chromosomes in the organism. Remember, the chromosome number is fixed to a species. How many in a human being? 46 chromosomes. If that number of 46 change, it is a chromosome mutation. It can also change the arrangement. And the example that we are going to study is Down syndrome. Now, Down syndrome is where we see when the baby is born, we see a flattened nose, upward slanting eyes, and interesting, the fingers are very short, especially the little finger. And there are this line right through the hand is also an interesting fact of Down syndrome. The toes that are widely apart, first and second toe, and lots of creases on the foot is a sign of Down syndrome. But what is different in the Down syndrome? It is pair number 21. If we look at the karyotype, you remember the karyotype is where all the chromosomes from the father and the mother are sorted from the largest to the smallest until we reach the 23rd pair. And now in Down syndrome, we see that pair 21 is not a pair, but we have 1, 2, 3. And therefore they say it is trisomy 21. They also call Down syndrome trisomy 21 because the mutation is visible in the arrangement of the pairs. It is not a pair in 21, but it is 3. What happens is during meiosis, we have the separation, and then they form two gametes. Actually, during meiosis, they form 4 haploid cells. But during this formation, the pair do not split. And where we have, the male has 44 plus the X and the Y chromosome in its genotype, and the female 44 plus XX. Now, they separate, and instead of having 22 plus X, there's 23 in the one chromosome, and in the other one, how many will there be? There will be 21 plus X. You see? But that one is not viable. So we ignore that one. That's not enough to have fertilization even taking place, or rarely take place. So if this is now a normal X chromosome, X sperm, and this is the other sperm with the Y. If we now have fertilization and this chromosome fertilizes the 23. Now we have 24. Can you see? That is an extra chromosome. So this will be a female, and this one will be a male. If this sperm fertilizes with that extra chromosome. That happens when we have meiosis in the female, in this case, meiosis in the female, instead of having the 23 pairs splitting one to that side, one to that side. During meiosis, it's not moving. 2 is moving to this side and none to that side. And then there are 21. So at chromosome 21. And there we see if there is an extra chromosome in the ovum. And that is more often the case, that there is more chromosomes in the ovum than in the sperm. The age of the mother can cause Down syndrome, the chance to have Down syndrome, to increase. If the extra chromosome is in the sperm cell, we will see that during the formation, we will have 23 plus X and 21, because these two must give me that number again, plus Y. You agree? And the female will be 22 plus X, 22 plus X. This one will give me now a female. If these two fertilize, I will have a female with Down syndrome. But I will not have a male with Down syndrome. There's no possibility to have a male with Down syndrome unless this 23 is now the one with the Y. You agree? If we have the Y there, I can have a male with Down syndrome. But if I have an X there, I will have a female with Down syndrome. I would like you, grade 12, to take your textbook and to check how they explain Down syndrome. Describe mutation as a source of variation as shown by Down syndrome. During the formation of the gametes, the sperm or the ova, 2 chromosomes of pair 21 fail to separate. The gamete has an extra chromosome. The egg or sperm can have now 24, 23 plus the autosome. Now, 23 plus 1 is 24. Okay, 24 instead of 23. After the zygote will form, 23 plus 22 will give us 47, 45, 46, 47. You see, this is equal to 47 because there we add the autosomes, the 6 chromosomes. Good. That is Down syndrome. And it is important that you know all the features of Down syndrome. Please do the attached question to see whether you do understand this or not. Read through this few pages, and then we have what can cause mutations to take place. Radiations and chemicals can cause mutations. Remember, cancer is a mutation. Why? Because the cell do not have the ability to stop dividing. It keeps on dividing, which means the message, the command for the cell to divide, to stop divide and start with its normal functioning is wrong. that will be a message from the genes that is wrong. So cancer is a mutation. When something can cause cancer, we say it's a carcinogen. You remember that word. But if we look at other things that can cause mutations, and it is a spontaneous reaction that takes place, we say mutagens are factors that cause mutations. UV rays, X-rays, gamma rays, alpha rays, beta rays. All those rays can cause the mutation. We know that the UV rays from the sun is the one that causes skin cancer. Then the tar in cigarettes can also be called a mutagen. Alcohol can be called a mutagen if it causes cancer in the stomach or wherever cancer is caused due to an overconsumption of alcohol. Now, we say the mutagen will not cause it, but it will increase the rate of the occurrence of a mutation. A mutagen will increase the rate of the occurrence. And with that, I want you now to please complete your worksheet. Interesting work today. Let's continue with variation. We are still busy with genotype, what is determined by the genes, and phenotype. Eventually, what you can observe in a person was in his genes. Sometimes we know the genes can be masked off because there's a recessive gene, and then it is not visible to see in the characteristics. But eventually, we can see a lot of variation in a specific species. When we look at this picture that we have here, is that we can see these birds are all Golden finches. They are of the same species, but we have a lot of color varieties between them. They are the same species, but there is a lot of differences between them. And then, of course, the human race. We know that there is so many variants in the humans. We must understand that with variation, we refer only to the differences between individuals of the same species. That is all that we are using when we use variance in when we use variation in a species. That is what we are referring to now. Now, as I said, this variation is caused by what do we see in an individual, what do we observe? And sometimes it is also not visible what is a person's blood type. But it can be A, B, O, or AB. But what causes that blood type? The blood type is caused by the genes. Nah? The blood type is caused by the genes. When we look at the phenotype, it can be caused by the genes, but also by the environment. Because I can have a nice dark skin, but it is because I am all day in the sun. That is environment. My genes tells me what is my blood group, and the environment cannot change my blood group. That is only by the genotype. We have two different variations. It can be continuous variation, which means there is no distinct category. It is smooth gradation. It can be caused by the genes and the environment, or by both. What am I talking about? I'm talking about height, skin color, intelligence, and there are many more variations. Let's look at first height. Here I have head of states, some not in the current running, but we can see that they start at 5'2" and they increase. It is a smooth gradation. There's not a distinct category. If you are head of a state, you are this length. No, it does not work like that. Another example, if we take the smallest feet in the school and we put then the next size, we will have a lot of feet next to each other. And they will just increase a tiny bit. Smooth gradation. No distinct category 4. Grade 5 feet are 12 centimeter. That's a distinct category. No distinct category for continuous variation. We also can have the same with eye color. We have a lot of different variations in eye color. And it's a smooth gradation. There I have skin color, fair to dark for male and female. It is a smooth gradation. You are not this color or this color or this color of skin. You can be anywhere in between. We have a lot of in between. But then the next type is discontinuous variation. So the first one was continuous variation, smooth gradation. No distinct categories. Discontinuous, distinct categories. There's no gradual change. There's no in-betweens. You either are A blood group or B blood group or AB or O. So there are 4 categories for blood groups, but you cannot be somewhere in between. Discontinuous, distinct categories. Blood groups, earlobes and attached or not attached, and gender are some of our examples. Now perhaps you cannot clearly see the ears there. I hope that I can quickly draw that so that you can see. There you see the ear, and there it continues, the neck continues. There is the neck and there is the ear. So we say they are unattached and these one are attached. If your ear is then linked to your face without having that, that is unattached ear lobes. Then tongue rolling or not. I wonder how many grade 12s can roll their tongues and how many cannot. And then we remember the fruit flies that we had in genetics. Some fruit flies had red eyes, some had white eyes. That's only genes. This is only your genes. This is only your genes. This is your genes. When a fruit fly has red eyes, it's the genes. When the pea has pink or red or white flowers, it's only the genes that cause that. So that is the second type there. Now, good. So here we have now a nice summary to help us to remember. Properties, continuous, no distinct category, no limit on the value. Now, the discontinuous, distinct category, there's no in-betweens. Here we have in-between. Smooth gradation. I hope you remember that. If you have to explain, that is the word you need to use. Distinct categories, no in-betweens. And we have some examples. What I want us here to use, this they call a normal curve. But I want us rather to use here the histogram. A histogram is where we draw the bar graphs close together. Now, we draw the bar graphs close together. Why do they touch each other? Because they are in-betweens. So we use a histogram instead of a line graph. And when we use distinct, we use a bar graph with definite spaces, because we have A blood group, we have B blood group, we have AB blood group, and no in-betweens. Genes and the environment, or genes or the environment plays a role here. Genes and the environment, or genes or the environment. And on this side, the genes plays the role there. So here I just try to explain. Please look. There we make an omission sign to show if we are not starting at 0. We have a range between that number and that number. I have 6 people. Between that number and that number, I have 9 people of that centimeters. Please make sure that you know how to draw a histogram. And I want you now to set a table of this length of a grade 12 class. Unfortunately, it is not you. It is you. And I add some other learners. Okay? Set this for me in a table. Draw a histogram. Okay, but we can still continue. And now, this variation that we are aware of, if we look at this female bodies, this variation, do you think it is gene? Do you think it is the environment that cause this variation? So is it the phenotypes, the genotypes? What are we looking at here? Of course, this differences between the two types of variation, continuous variation, we said, is caused by the genes and or the environment, right? Discontinuous variation is caused by only the genes. So, what influence environment? What influence variation? What influence variation? Our variation is influenced by our genes, but it is also influenced by our environment. Let's start again. We have 2 types of variation. Continuous and discontinuous. But what influence this two types? It is our genetics and our environment that will influence how the variation will play out. And I want us to take us again, revision I know, but let's look at the genotype. Please remember, my genotype is being made up of my gene. And each gene has 2 alleles, except if it is an X-linked. So there I see the father and the mother's chromosome inside a cell. During DNA replication, it is doubled, and then we know that crossing over happens. With crossing over, look what's happening. The genetic material mix up between those 4 chromatids. And eventually, look at, if there was no crossing over, that's only from this person's dad, that's only from this person's mom. But it does not happen like that. We have crossing over, and then we have this separating, and they separate in different shapes and sizes, and therefore already to form the gametes during meiosis. Please remember, I already see a huge difference in how my offspring will look from the mother's or the father's cells. You agree? And here I just have a picture to show. With crossing over, there I have now father, mother, 2 chromosomes. They mix up and link, and all the sizes change. And eventually, look how many options are there in for making the gametes. And that is where I already have my crossing over, and the end of the gamete. In the end, the gamete will not have exactly the same combination of genetic material that was in the mother cell. Okay, and then we must remember what happened during fertilization. I just want to go back to that slide of mine. The genotype is made up of 2 alleles. The meiosis causes a mix-up of the alleles between the person that's body is forming the gametes, parents. And what is in the parents? 2 other parents. Then, fertilization. We know there are a million of different sperms. And one of those, with its specific gamete content, will eventually end up and fertilize one of those options that happened in the ovaries. So all that eventually makes a huge variety possible in the offspring. Then when we look at the environmental variation, we say it is acquired characteristics. And that brings us to the environment. What can happen in the environment? Let's make a list. The type of food, the amount of water, that a plant gets. But here, what is this? This is a beautiful bonsai of the camel thorn tree. And what happened here is it was pruned since a very young age to shape it in a specific form. Amount of sunshine. All this eventually cause a specific outcome.
Hi everyone, we are from the Giraffe Conservation Foundation. Our team protects and supports the safety of giraffe in 15 African countries. Together, we are going to share and explore lots of interesting environmental facts with you. I'm Audi.
I'm Martha.
I'm Naomi. Welcome to episode 8 of Environmental Learning of the Giraffe Conservation Foundation.
Today we are going to spend time with all things wild. Wildlife refers to all living things that live and grow in a natural environment in the wild.
Wildlife in a natural environment can be found in many places around the world. Most often they live in protected areas such as national parks, game reserves, and private game farms.
In other parts of the world, like in Namibia, wildlife also share their living space with people.
There are many different types of wild animals and they all belong in particular groups. There are 3 ways to group wild animals. They can be grouped according to what they eat, their feeding behavior, and their active periods.
Let's start grouping animals according to what they eat. Herbivores eat only plants, for example elephants and giraffes.
Carnivores eat meat, and that means they eat other animals. And the examples are such as lions, cheetahs, and leopards.
Omnivores eat plants and meat, for example, baboons and jackals.
Insectivores eat insects, for example, pangolins and geckos.
Detritivores eat decomposed dead animals and plants as well as dung. And the examples are millipedes, maggots, and dung beetles.
Now that we know what herbivores and carnivores eat, let's go into more detail about their feeding behavior.
There are 2 different kinds of herbivores, grazers and browsers. Grazers mainly eat grass, but also other soft herbs, for example, springboks and wildebeests.
Browsers eat leaves, fruits, and flowers from the bushes and trees. And the examples are kudus and giraffes.
Predators kill, hunt, and eat other animals, for example, lions and leopards.
Scavengers eat dead remains of other animals, for example, hyenas and vultures. Now do not get confused. Both predators and scavengers are carnivores because they eat meat. The difference is predators hunt and kill other animals, while scavengers do not hunt and kill. They eat remains of animals that have already been killed or have died of a natural death.
Now let's look how wild animals are grouped according to their active periods. Many animals are diurnal, and that means they are mainly active during the day. At night, they sleep or rest. And the examples of these are giraffe and warthog.
Some are nocturnal. This means that they are mainly active at night. Examples are leopards, bats, and tigers.
While others are cathemeral, and this means that they are active both during the day and at night. Examples of these are lions and elephants.
The well-being of wildlife is threatened by a lot of different kinds of human activities. The main threat to wildlife is expansion of human population. As the human population grows, more land is needed to grow food and build houses for town and city development. And as a result, space available for animals and plants become less and less.
In areas where wild animals and people share the same space, it can happen that wild animals and people compete with each other. Infrastructure such as roads and railways divide wildlife habitats into smaller areas. As a result, wild animals can no longer move freely without danger. Illegal hunting or poaching is a real threat to wildlife.
Illegal harvesting of wood also reduces wildlife habitat. Plants and animals also face natural threats, and these threats are climate change, global warming, and drought, which result from change of climate.
The main reason for conserving wildlife is so that they can continue to exist. By this, the future generations of people will have a chance to live in a natural world and enjoy all the amazing things that live in it.
Conserving wildlife keeps the world in balance. Each plant and each animal plays an important role in maintaining health ecosystems and contributes to the quality of our lives. If they disappear, it will have consequences on all of us.
Wildlife attracts a lot of tourists. The tourist industry contributes to the continuous existence of wildlife and create employment for people.
It is extremely important that we protect and support our wildlife. We can do this by educating people and raising awareness on the importance of wildlife.
Maintaining and restoring them to their natural habitats, reintroducing them to their safer natural environment.
Protecting them and supporting them where they are already. Saving our world wildlife is very important to all of us now and for our future generations. So stick your necks out tall and long for giraffes and for all other organisms that are natural and wild.
We have come to the end of our time together today. But before we say goodbye, we'd love to leave you an action that you can do by yourself, with your family, or with your friends. Goodbye, everyone. And remember, every little step in the right direction counts.
The coronavirus that causes COVID-19 has changed since the pandemic started. So does this mean the virus has become more dangerous? Scientists have discovered thousands of mutations to the SARS-CoV-2 coronavirus. Most mutations only lead to small changes, but scientists believe one, called D614G, may have made it easier for the virus to infect human cells. This mutation most likely emerged after the initial outbreak in China, and it spread fast after the virus arrived in Europe. By the end of June, the D614G mutation was found in almost all samples of the virus worldwide. Scientists believe this is due to amino acid changes in the virus's spike proteins, improving its ability to infect human cells. Some studies have found more spikes on viruses with this mutation. These spikes latch onto receptors on the surface of our cells. They then transfer the virus's genetic material, called RNA, inside. Being inside the host cell allows the virus to make many copies of itself. Random errors sometimes happen in the copying process, which cause mutations. Although the full effects of D614G are still unclear, some studies suggest that the mutated virus might be more transmissible than the original. This could mean more people getting sick and at a faster rate. Despite this, there isn't any evidence that the disease itself has become more severe. Scientists are worried about mutations possibly increasing the risk of reinfection. Hong Kong reported what could be the first case of virus reinfection on the 24th of August. A healthy man in his 30s is thought to have caught the virus again 4.5 months after he first got sick. During this time, mutations developed and new variants of the virus emerged. Some experts believe these changes may have made reinfections more likely. Mutations are common over the course of a pandemic. New flu vaccines are needed every winter to keep up with genetic changes to the flu virus. If a coronavirus vaccine is found, it could require regular booster jabs. Mutations won't happen overnight, and the coronavirus is mutating slower than the flu virus. But it's important for scientists to keep a close eye on how the virus changes over time. Could we be hearing more about other mutations in the future?
Omba Arts Trust is not just an organization for me. I think of it in the form of a person, a mother and a friend I have walked and grew with. A friend whom I've traveled many remote and dusty roads. A sister who provided so much needed support during times of fear and uncertainty. There is a saying in Damara for people such as Omba, which is which simply means someone who will, with love, exchange your lean piece of meat with their fat one. It might sound simple when translated, but when used towards a loved and appreciated person, it gives such honor. It describes a person who will do everything to make sure you are taken care of. Besides, we all know how the Damaras love their meat.
Ja, dit sê maar ja. Goed. Jy nou moet 'n bietjie meer reg wees, né? Soos die boere hulle mielies plant. Ja.
I have seen Omba grow and turn into a light of hope for Namibians all over Namibia. I have seen women and men grow as individuals, becoming respected members of their communities, but also valuable providers of their families. I have seen people with no hope in their eyes light up when they see Omba arrive to fulfill a promise which was made and which they did not believe will be kept. I have seen women dance and sing for hours until the dust from their feet covers us all because the old friend has arrived. I've seen women standing up in front of a large number of people and express their hopes and dreams. I've seen people who have never left their village travel hours and hours to other countries and share their culture and experiences. I have seen children grow and become young women and men before my very eyes. Omba has been the sister holding hands throughout it all. Omba has built valuable and strong relationship with communities, empowering both women and men, but the impact on women can be seen and felt most sincerely. The work with the women has such a ripple effect all over the whole village, most especially the young. Omba has taught them to fish. As times grow tougher and much more difficult, money becomes scarce. Environment around us changes and nothing will ever be the same again. In times like this is when we realize the value of a friend like Omba. It provides hope, constant support, reliable income, and keeping us all together, keeping our pots filled and our fires burning. A motivation to do better, live better, and expect better. If one day the unthinkable happens and Omba is no more, it will be a sad time for our Namibian rural population. Therefore, let's keep the light burning at all cost.
Ons die Sanmense is die eerste mense in die wêreld wat vandag agter gaan staan. Omba is nou die water wat vir ons nat maak, dat onze wortels mooi groeien, dat ons vruchten kan brengen, kos kan brengen. Omba gee vir ons werk. En van daaraf lewe ons.
It's just an amazing story that ostrich eggshell beads, 72,000 years ago, the ancestors of the San were making ostrich eggshell beads, and today in the 2020s, Junko women are still making those beads pretty much in the same way. And the work has evolved from fairly traditional products, this probably would have been worn like this, into something as sophisticated and contemporary as this necklace from the waterfall range. So here we are bringing the necklace to this gallery in Windhoek, and it then gets sold to customers in Europe and America, and generating income for women, Junko women back in the Kalahari. So it's just an amazing thread of history that goes to this ostrich egg, to this contemporary jewellery. This basket I absolutely love. It's a finely woven basket in natural dyes. This asymmetry is very unique to Namibian baskets, and we've been working with these women, doing workshops and helping them evolve into these incredibly fine baskets. We've worked with the women developing different palettes, and there's another one with a slightly more yellow palette, but considered are the best baskets in Africa. What we're showing you in this short video is really a long journey of working together with rural communities, reviving ancient traditions, turning them into contemporary world-class products, and supporting the livelihoods of over 400 artisans. Even during COVID-19, we've been helping our sand communities with food and distribution of tippy taps. What we need to do now is to honour the trust that has been put in us over the last 30 years, to support marginalised sand communities and the livelihoods of rural women around Namibia. Your donation will help us get our 400 artisans back to work as soon as possible so that they can feed their families. The lives of their 2,000 family members will directly benefit from this. Please help us raise $50,000 by contributing to the Umba Artisan C19 Support Fund. No matter how big or small, this will make a difference in the lives of our artisans during COVID-19. Thank you.